Most of us have heard of the plague of locusts. But did you know that to this day, desert locust plagues still threaten the food security of millions? In this episode, Yannick Günzel and Inga Petelski will tell us all about how the population density and social order within a locust population can transform shy, solitarious locusts into hungry, gregarious ones who are ready to march with millions of their friends to search for the next meal. Hello and welcome to In the Swarm, where we dive into the minds behind the hive. I'm very, very happy to welcome Yannick Günzel and Inga Petelski. Welcome to the podcast, and I'm so happy to have you both here.
Thank you.
Yeah, thanks for having us here.
Hi, would you like to quickly introduce yourself? Maybe Yannick first?
Yeah, I'm Yannick Günzel. I'm here as doctoral student and affiliated with the cluster basically from the beginning of my PhD. And I'm really obsessed with locust decision-making in social contexts.
I'm Inga Petelski. I'm also a PhD student in the lab of Einat Kazan, and I'm working on locusts, have been working on cockroaches a lot, and I'm currently on maternal leave, entertaining my little son at home.
Oh, perfect. Can you tell us briefly how you guys basically find yourself in Konstanz and why locusts and why this topic in general? Did you have this interest right from the very beginning, or how did you find yourself into this field?
So I did my bachelor's and master's in Bielefeld, and from early on I could focus on neuronal mechanisms underlying behavior and evolution in general. And throughout those studies, I tapped into monkey and fish science, but all the time I came back to insects. So for my master thesis, I looked into motor control of stick insects. And then I saw the project on collective decision-making locusts here in Konstanz. And actually, I applied 1 month too late. And I wrote to Einat like, hey, this project sounds fascinating. I'm so intrigued. This fits perfectly my interests. So I applied and she said, okay, fair enough, come. And yeah, so this is how I ended up here in Konstanz and started my PhD in 2019.
Oh, perfect. So no deadlines are hard deadlines. If you want it enough, just keep trying. And Inga?
So I've always been very fascinated by animals and their decisions and their behavior, especially actually in deep sea creatures and flying animals. So I did then my bachelor thesis at the ornithology in Radovce with mallards. I then moved towards flies and did neurobiology in flies for my master's. And I did a little detour and did research at a marine conservation society in the Seychelles Island to then come back to the lab of Einat again and do the neurobiology PhD, which I'm currently still doing.
Oh, wow. The vibe I'm getting from both of you is that as long as you're interested in animals, insects, whatever, you can study them. How is it for you? Because Inga, you went from kind of marine biology straight into insects. And for you, Yannick, you also did this curly-wordy path. You did animals, you did monkeys and all of these things. And now both of you are doing locusts. How does the animal model, how does that inform you? Because if you ask me, it's kind of obvious to me that if you're a biologist who's interested in animals in general, you kind of have this overall fascinations in how things work or how different animals / insects, etc. work anyway. How does that kind of inform you? Because, you kind of we're interested in everything and now locusts.
I guess as a researcher, the good thing about it is that you can cherry pick the topics which excite you the most. So you just go into one direction and then really find out whatever you want to find out and whatever interests you in this topic. But then if you're interested into another species, for example, or other mechanisms, you can always move on.
Yeah, so actually my path was not as curvy as it might sound. So I was as a student assistant in an insect lab from the end of my bachelor's onwards. And I think the cherry picking is a great example of what I did. So I had my cherry and I was kind of probing the nearby trees of maybe there are better cherries available. But always the insect cherry was the one I got stuck with. The locust, I think, is just fascinating because it has so many different aspects of what you could study in a single animal.
What got you first interested in them, really?
Yeah, so it was the cluster's first birthday party, and we were all coming together. Everything was super new for basically everyone. And Anna just got the news on her phone that there's a big outbreak of locusts going on at the Horn of Africa. And we decided we have to go there. This is such a once-in-a-lifetime opportunity to investigate the swarm in nature. So at the birthday party, all of us locust researchers came together, and just on the spot, we decided we have to go there next month. So we wrote a small cluster project grant to get funded to go to Kenya. And really 1 month after the birthday party, we were in the field looking at the locusts, how they swarm, how they cover the whole plains of where we've been. And this was, just when you think about it, such an ad hoc experience. It was super cool.
Yeah, awesome. Can you describe for us a little bit? Because, okay, you got on the news on your phone, there's a swarm going on at the Horn of Africa. And you're like, okay, we have to go. I don't even know how I would arrange the trip. You know what I mean? It's like I would just try to find accommodation for myself, for my partner, get a flight, and then go and then, do whatever I want. But then you have this big, very time sensitive, I would imagine, incident or natural phenomena going on. How do you even take the first step arranging that? Like, who do you need to contact? Can you explain that a little bit?
It was a bit overwhelming in the beginning, what we had to bring to the field. If we need mosquito nets, GPS, some dye to mark the animals, and scissors and tweezers and all those little things which we had to remember. And then we got in contact with Zell, with the Max Planck there. And they have this station, the
Impala Research Center. Yeah.
It's located in Kenya. So we went there. That was our first stop. And there we could talk to other researchers and talk to them about what we needed in the field. We wanted to stay over a month, but then our trip was a bit shortened due to Corona, unfortunately.
Good old COVID pandemic. Yeah.
So the actual adventure then just started after we left the Impala Research Center, because we went there. So this was our base station basically, but there were no locusts around, and we had some local contacts from Kenya, and they had some GPS coordinates of where they have spotted the swarm before, but this was out of our reach, also for security reasons. This is why we got Max car after all, to explore central northern Kenya areas ourselves. So the four of us, Inga, Felix Oberhauser, and Einat, and I, we hopped in the car, loaded the flatbed of the car with all of our equipment, put a cover over, and just drove, hoping for the best, basically. And every once in a while, we got out of the car looking for locust poop, because
first of all, we couldn't find anything. So we thought the whole Kenya is covered in locusts, and we only found poop. And we're a bit frustrated in the beginning.
And we also had to ask the locals, but we didn't speak the language. So what we were doing is we drew small locust cartoons on pieces of paper, and whenever we stopped, we just showed those cartoons to people, and we were asking like, hey, where's the locust? Can you help us? And eventually a Maasai warrior who was traveling between villages just pointed in a general direction towards this village where he was coming from. And this is then where we finally found the locust swarm, and we kept coming back to this place for several days because every day there was more locusts coming. And just imagine you look around yourself and the whole ground is moving throughout the whole day for days. This was truly mind-blowing because after we found them, they were just there and they just ate so much. And the locals described to us that the environment used to be very green, very bushy, but it was just like scrubs left, and interestingly, toxic plants that the locals used to treat malaria that also the locusts didn't touch, which was really also interesting to us to learn more about the local vegetation and also the local dynamics. Yeah, so then the real adventure just started.
Yeah, we then cooperated with some of the Maasai. They always showed us where to go. It was really helpful to work together with them.
Oh, wow. That's super cool. So I understand that a swarm is not completely stationary. It has to be dynamic to a certain extent. But you did say that you had to go back like day after day to this general location to really kind of study them. So they're moving, but at a slower pace, I'm presuming.
Yeah, and there were more and more swarms coming and emerging then. So the weather was very favorable to them. So there was a lot of rain before. Suddenly a lot of vegetation grew. So the locusts had favorable breeding conditions. So they were all laying eggs, and all those eggs were emerging around the same time. Then when all the locusts were in contact with each other, this then gives rise to serotonin in the brain of the locusts, and they then become gregarious. So the fascinating fact about locusts is that they either exist as a solitary animal, they're very shy, cryptic, they're green like the bushes where they feed on, and they avoid conspecifics apart from mates. However, when there are loads of locusts, then they touch each other, and then they start becoming gregarious. They are bright yellow and black, and when they're adult, they're bright pink. Whereas the solitary animals, when they're adult, they are more brown like the sand. And those gregarious locusts, that was what we were looking for, and we also found. And more locusts were then emerging, and we could drive around, and then we found more and more swarms wherever we came. Yeah, one swarm of 1 square kilometer can house around 80 million animals, and they eat as much as 380,000 people in a day.
Yeah, I think here it's also important to add that now when we were talking about the locust swarm, it's actually not the adults we were looking for, but the juveniles that don't fly yet. So they don't have proper wings, they just have wing buds, like tiny wings. And this is then also the stage where you can actually fight them, because they are less mobile than an adult flying swarm, which could migrate roughly 1,000 kilometers within a week when they fly with the wind. The juveniles, on the other hand, they have to walk. And this is then also in the end what we recorded.
Mm-hmm. So I guess with the juveniles, since they have to march, if you're gonna do something about the swarm, like curtail them or try to eradicate them for whatever reason, maybe if you're local and you're seeing, all your crops being destroyed all at once, this would be the stage where you could do something about it.
Exactly. And there the common practice is that you spray chemicals to just kill all the animals, literally all the animals, because it's not very locust specific. Exactly. And also the chemicals you apply, they stay in the food chain because they stick to the plants, and they tell the locals to not have their animals grazing in those areas for some time. But of course, who knows how long those chemicals are staying.
Yeah, it was very sad to see, because we knew that those are neuroinhibitors which they're spraying. And then we saw on the next day already the locals had their animals grazing over there on those areas, because those were the only areas where there was some vegetation left. Mm-hmm. So they had to do that. Yeah. So nowadays there are also some biopesticides, some fungus-based pesticides, but they're not using it too much yet. Yeah. And I don't know about how effective this is. Mm-hmm.
Mm-hmm. But I'm guessing that once they reach like a mature state, it's not much that you can do anymore in terms of making the swarm go away.
Yeah, some birds and reptiles are eating them, but
not at the size of the
Yes,
swarm, right?
not at this sudden size.
In order for a juvenile to go from a solitary state to gregarious, they have to touch each other in a certain way, right? 1, what does it look like? 2, and how long would that really take in terms of like, if I touch you for like 5 minutes and I'm like, I guess I have to become gregarious now. Like, how does that work?
It has been reported already that we also saw that 4 hours are already enough for them to change their behavior. If you put 8 locusts in a little box, then they walk over each other. So this is already enough, especially the hind legs are important. And once they are touched by the other ones, just by crawling over each other and by a certain density of locusts, that automatically happens. Mm-hmm. And then, they start changing their behavior, and also within the brain, neuronal activity is changing.
Mm-hmm. And how long would this last, this gregarious state? Because I'm wondering if there is some sort of scientific magical way of changing the swarm behavior from gregarious back to solitary. Wouldn't that solve the problem? Then we wouldn't have to use pesticides that kills everything, and destroys the food chain and stuff like that.
So that's very interesting. So the species we're looking at is the desert locust, Schistocerca gregaria, but there are also different locust species. And different species have different dynamics of how long it takes for them to turn gregarious or to turn back to solitarious states. And our species, they become gregarious super easily. As Inga said, it takes about 4 hours, and their behavior completely changed. But then turning back to a solitarious state takes way longer for the desert locusts. But other Schistocerca species, for them it's the opposite. And this is also dependent on the different habitats they can be found in.
So how does it work that eventually the swarm kind of dies off or disperse? Because otherwise, this is like a huge problem. It just keeps on going, it keeps on destroying different habitats.
One factor is the temperature. So the locusts thrive at around 30 to 40 degrees. So once it's too cold, they cannot survive anymore. And also at one point, they don't find enough vegetation anymore to feed on. And then the plague slowly decays.
Can you tell me about how you use olfaction to study this swarm of locusts?
Our study of olfaction reaches back to the different phases of the animal, actually. So we have the gregarious and the solitarious phase with completely different lifestyles, basically. And this also changes the different information classes the animals have available for their foraging decisions, for example. So the solitarious phase basically has no social information, only its private information about the environment, whereas the gregarious phase has both information classes. So what does it mean to have both social and personal information? And I always compare this, for example, to the Konstanz Weinfest, when you would like to find the best wine available. And on the one hand, you maybe remember from last year which wine stand was the best one, and maybe you're more tempted to go there. But on the other hand, you could also look at the queue length, because this also should inform you about which wine is the best one, because more people should queue for the better wine. And now you could integrate those different information for optimal wine foraging, so to say. So this is also what we did with the locusts after all. Obviously not with wine, but with blackberry leaves. So we presented them different options with different classes of information. So we had an option that contained only food. We had an option that contained only animals. And we had an option with animals and food and an empty control. So we tested both gregarious and solitarious animals behaviorally, how they choose those different options. And we saw that the gregarious animal loves the combination of food and friends, basically. The solitarious animal, on the other hand, only likes the leaves. And then we went further and modulated how they can sense those different options, either by just seeing them or by just smelling them. And together with mathematical models, we identified that the sense of smell was really important for them to make this decision reliably. And this is then why we continued with investigating their olfactory sense and how they process relevant information in their brain.
Cool. I was trying to kind of tie your work in the field with your work in the lab. Did your field trip actually make you think about the olfaction side of things, or did you have that idea before? How did it all come together? I'm curious about that.
So actually, I had some odors with me, and I wanted to do some experiments where I redirect the whole swarm, or the marching band, so that they walk into another direction, but they somehow didn't care. And somehow first we thought, okay, the odors, they don't really play a role. So for their marching, when they're marching all together, then it wasn't that important. But we could then see that when there was food available, somehow the whole swarm stopped. And they were all distributing and searching for food all together. So, they were waggling around with their antennae. So, because locusts, they don't have noses. They have antennae, which they use to smell and detect odors. And we could see that they were actively moving them around to search for food. And then we thought, okay, let's tackle this and see if they really need more vision and look around for food, or if they use their sense of smell.
So actually, locust swarms are really smelly. And so when you ever have been to our breeding colony, you smell them super strongly. And this is also what kind of motivated our story a bit, is that within the smelly locust swarm, individual animals still have to be able to smell the food around them, although it might be masked by the smell of the conspecifics around them.
Circling back to the study that you conducted in the lab, so what I have in my brain is that you have different boxes. And the first box is empty. There's nothing in there. Second box is food. Third box is food and friends. But what I'm actually getting a hint from you here is that when these poor locusts who had to take part in your experiment, when they go into the box that they think is the best, they don't find food and friends, it's just smell. So can you tell me a bit more about how you replicate these smells?
So we had this behavioral arena, which you just talked about, and there actually we just monitored if they go close to those boxes. And in those boxes, we had friends and leaves and all those things in there. And we then wanted to look into their brain with calcium imaging and see how those odors are then processed in the brain of those locusts.
Ah, okay. So there are two stages to the experiment. The first one, you have this big behavioral arena. You let your locusts out and just basically monitor which box they go closest to.
Yes.
And then based on that, since now you want to look at, okay, what's happening inside the locust brain, you would just expose them to these different smells that you would manufacture yourself. And then you use calcium imaging to see what's going on inside the brain.
Yes,
right?
We always had single individuals in the arena, either a gregarious one or a solitarious one.
Okay, so just one at a time.
Yes, always one at a time. And we were wondering why they have those very different decisions. And then we wanted to look into their brain and see if already at the first processing center for olfaction, if there's already a change which we can record or if we can see something that can explain to us their very different decisions.
Tell me more about calcium imaging. Like, what is it? How do you use it? What does it tell you?
So calcium imaging is a really nice method to look into the brain and the activity within the brain of animals. You can either do it in cell cultures or you can do it in living animals. We had to inject a calcium indicator into the locust brain in the area which we wanted to record in, which is the first processing center. It's the antennal lobe of the locust. And whenever there's activity in the brain, a neuron does fire via action potential with a membrane depolarization. And then we have calcium influx. This calcium is flowing into the cell and then binds onto our indicator. There's a conformational change. And then we have an increase in fluorescence. So we always, when there's action potential, so there's activity in a neuron, then we have a firing or lighting up of our calcium indicator. So we have like a firework in the brain of the locust wherever there is some activity going on.
So would you be looking for specific patterns in these fireworks, or like do you have already a working hypothesis that because of this we expect these patterns, or how does it work for you?
So the problem is that the antennal lobe houses those little processing units, and usually there are just a few of those units, up to like 200, for example, in the cockroach. But in the locust, they have over 2,000 of those processing units. So it was really hard to make sense out of those firing patterns. But magically, Yannick managed to do that.
The cool thing about calcium imaging is that you can observe network spanning dynamics of the calcium influx, basically. Previously, because of the complex architecture of the locust antennal lobe, previous studies only poked individual cells and recorded from them one after another. And now, with our approach, we could monitor almost all projection neurons, which are the output neurons of the antennal lobe, over time, how they were processing the different stimuli, the different odorants we gave to them. And on the one hand, this allowed us to observe changes in magnitude of how neurons responded, how strongly they responded. And there we saw that the mixture of the smell of food and friends elicited really strong responses in the gregarious animal, but not in the solitarious one. And since we were able to observe the different dynamics of how neurons responded, we could also nail this down to certain response motives that acted synergistically, meaning that it's stronger than you would expect from just addition. So we observed those synergistic effects in the gregarious, but not as much in the solitarious animal. And this really allowed us to connect our behavioral observations of gregarious animals choosing the mixture of friends and food to our imaging experiments of where the response magnitude was strongly synergistic for the smell of food and friends.
Okay, so from what I'm hearing about this calcium imaging is that when you compare locusts between two different states, solitary and gregarious, you're getting really very different firing patterns, like really very different looking fireworks that you can see in these poor locusts' brain. How do we get these different patterns, Yannick?
So this is a super interesting and also fascinating question to me, because this is exactly what we are trying to nail down now. What is happening in the brain that allows the animal to display those different behavioral choices. And something that is unlikely happening is that it's a completely rewiring of the brain, because the animals we are using are first generation solitarious animals. And this means we basically take them after they hatched in our gregarious faculty, and then we transfer them in solitary confinement, basically. And then they grow to becoming a green solitarious animal.
So this got me thinking a bit more generally in terms of insect olfaction, because I feel like if you ask a general layperson to compare between, I don't know, a dog and a beetle, they might say that, or they might have a personal take that maybe a dog is more dependent on its sense of smell as compared to a beetle, for example. And I'm getting a feeling that that might be completely wrong.
Yeah, so actually you would be surprised by how important the sense of smell is to different insect species. And just think about the fruit flies invading your kitchen because they smell your rotting banana, for example, or mosquitoes that love your smell, and that's why they eat you alive, basically. But also for us humans, the sense of smell is crucial. And I mean, humans don't think about olfaction on a daily basis. And just when you lose your sense of smell, you just realize how important it was to you.
So now that I've been informed and educated about this very, very fascinating study, please tell me about the process of getting this published.
It was a very long process. So it sounds very clear and very systematic, but getting this whole calcium imaging to work was a nightmare. It took years, and it was really complicated to finally get signals in the brain, as there hasn't been a really established protocol for it. Yeah, we were poking in all different parts of the brain to see if the dye travels in the correct neurons, and we then for a long time didn't have nice responses. We changed dyes and had to change the whole procedure of opening up the brain so that the locust is happy and alive.
Okay.
We had to change the temperature, so it was really a really long process. Yeah, I've been doing it in the cockroach already, and I did it in a fruit fly as well. But the locust, with all those tiny processing units, which we wanted to first make sense out of it and also have them stained in the first place, was a nightmare, actually, yes.
I was a bit more lucky in this regard, because I joined the project later, and I was basically sitting in my ivory tower waiting for the data and then to analyze it and then try to make sense of it. And I think in this regard, Inga and I were a great team, that she, with her magical hands, got all the data, and I was then sitting at the computer trying to really dive into what she has recorded. Since it's such a complicated process, it's really important to celebrate all different stages of your project with the whole team, preferably. So we have the tradition that whenever there's a milestone achieved, like submission of a new paper, we come together and celebrate this. If it's rejected, because this happens so often and no one really talks about it, we just come together and motivate ourselves. If it's under review, we come together and just really try to keep the whole group happy, because those spikes of happiness is really what you have to feed on, because science is complicated and you really have to show endurance. I mean, we are all super fascinated and curious, but sometimes just things don't work out. And then it's also important to remember that others struggle in similar ways, and then coming together, like reassuring, like, hey, that's all normal, is really helpful to stay motivated even during challenging times.
Yeah. One of the reasons why I also wanted to talk about this, from your first step to the publication process, it's, as you said, it can take so long. There can be so many happy moments and also nightmare moments, as you said, Inga. But yeah, is it out? Is the paper out? Can we read it somewhere?
Yes, it's out. Finally.
Yes, finally it's out. Yay!
It's out now in Nature Communications. And
wow, congrats.
It's an open access journal, so everyone can read it, which is also very important to us.
This is super amazing. And now let's take a detour back to you guys as researchers. How are you finding it? You're both PhD students. Obviously, the locusts are kind of the most important thing going on in your life, but I'm sure there there are also other more important things in your life as well. And how are you juggling or balancing through everything?
So academia is both. It's amazing and really tough at the same time, I think. And I think the more you experience it, the more extreme both aspects become. On the one hand, we have opportunities of going on fieldwork, but also on international conferences, meeting international colleagues all over again, completely nerd out with them about our weird insects, for example. On the other hand, it's challenging. It's insecure job opportunities. It's always very short-term contracts.
It's not something that is all shiny happy. You always have to try to be one step ahead, try to apply for jobs, apply for grants and proposals and things. And to be honest, I don't think it's negative. It's just how it is. We wish it were better, but it's also what it is.
But it's also great. So after my PhD, I want to continue as a postdoc investigating insects. I mean, they're just fascinating. And the closer you look, the more complicated they are, which makes them even more interesting.
Amazing. Inga?
Yeah, I think this study also showed us that teamwork is especially important, because I had some major drawbacks during my whole PhD. So it was really cool. I figured out this whole calcium imaging, but then Yannick made sense out of my data, and then we had other colleagues who helped, and it was so important to work together, which was, an amazing experience to have.
Especially Einat was a really great supervisor in this regard, always supporting us. And she always kept us all together and motivated.
So now it's even more special that it's finally out now.
Exactly,
Yes.
for the whole team.
Yes.
Amazing. Yeah, but this is something that I hear again and again. Yes, it is about the scientific content, but it's also about the people that you get to work with, the people that you get to engage with within, the science community in general. Really having people who support you, that you can support in return, it really gives you and your coworkers really special kind of strength, I would say. And it makes you better, and it makes you think, differently in a certain way. And I find that that's what really amazing about science, that you get to, in a way, choose your coworkers and your colleagues, because you're kind of all united by your fascination on similar projects. And then you get to kind of speak the same language. And I find that really, really nice. And I'm so glad to hear that you have really, really good supervisor experience, because also that can happen, that your supervisor just are not able to give you the support that you need, and then people react as if it's a personal failure, that, oh, I'm not good enough. But actually, it could very well be that you're just not getting the support you need. Anyway, so Inga, you might jump back into your deep sea creatures.
Yes, let's see. But first, finishing the PhD after the maternity leave, and then I might, let's see.
Yeah, so I want to stick with insects, and I would like to leave Germany, because I've been here for my whole life now. And now I think it's, time for a small adventure. And locusts are still fascinating. And with this regard, I really dream of having transgenic locust lines.
What does that mean?
So this means that we can genetically modify different cells, that we don't have to inject the calcium-sensitive dye, but that it's genetically encoded, which would make so many things way easier to do in the end. And I really hope that the field develops in the direction that we also have other transgenic insects. And so this is where I see myself heading towards too.
Awesome. Best of luck. And I think that's also one really nice thing about science/academia, is that you're really not bound to one place or one country. You can really, take your interests or your research repertoire and go anywhere else in the world where there are people who are interested in looking at similar topics or questions. One last question that I always like to end with. It's what I call pep talk for academics. How do you know that academia is right for you?
I sometimes feel I'm working a bit like a spy, because I'm secretly observing the animals. So we try to check what they're doing. We look into their brains, and we want to somehow translate it, understand what they do, and somehow make sense, so that it makes sense for us as well. So for me, this is a really, really exciting part. And spying on the animals and really understanding their goals are
on a mission.
Yeah, so this is really the most exciting part.
So when I realized that I really have to continue doing research, is that I talk so often at so random occasions about locusts, like be it a doctor's appointment or just with my family. It's so weird that eventually my mind goes back to research and figuring out what the locust is doing. That I think this fascination is what really motivates me. And I think this is, if you feel obsessed with a topic and you just want to know what the heck is going on, then I think this is, what you should do.
Yeah, if you walk around outside and you constantly think, oh, what is this animal doing here? And why is it doing this? Then
yes, immediately take a picture.
Already, yes.
You're there.
Yes.
You're with us.
You're already on the path towards biology, at least, yeah.
Well, great. Thank you so much for your time. It's been such a lovely time chatting with you both. Best of luck with the rest of your PhD. I'm sure you don't have much left to do, but just wrap everything up in a nice little bow and send it off. And thank you again for tuning in to In the Swarm, and we'll see you next time. Bye.
Thanks. What a pleasure.